Okay, so good evening, everyone. So today is the last session or a problem solving session for this NPTEL course, Genetic Engineering Theory and Application. In week 12, we mainly revised all the concepts which we have been discussing or which we have been learning in throughout this course. So from the week 1 module to the week 11 module, all the concepts that were covered were revised in week 12. So today also we will quickly try to revise all the concepts, the main concepts that were covered in this course. Right, and if you have any doubts, please do ask. So if you have to summarize what are the topics that were covered in this course, genetic engineering. So we started learning about the structure of a cell. So what are the different types of cells? What is the difference? So a prokaryotic, how does a prokaryotic cell differ from an eukaryotic cell? Then in an eukaryotic cell, how a plant cell is different from an animal cell. Then in prokaryotic cell also we looked into the structure of the gram-positive and gram-negative bacteria, right? The gram staining method we learned. Then we learned in brief about the cell metabolism, the metabolic pathway, glycolysis, Krebs cycle, electron transport chain. And then we started understanding about then we learned about then we moved into the main concepts of genetic engineering. What are the restriction enzymes? What are the other than restriction enzyme? What are the different enzymes which are used in molecular cloning? Right. Then we learned about the vectors. What are the different cloning vectors, expression vector? What makes an expression vector different from a cloning vector? Then once we have cloned the gene, how that cloned gene is transformed inside a host cell. How do we select a host cell? Um, how do we screen for the host cell which has taken up the recombinant plasmid, right? So these are the upstream processes that is involved in genetic engineering, and then the downstream processes is involved with expression of the gene. So how do we express the expression of a gene means the gene will be transcribed into mRNA and then the mRNA will be translated into protein. So how do we express a protein in a host cell? And then after the protein is expressed, how can we isolate that protein? How can we extract that protein from the cell? After we extract the protein from the cell, how do we purify that protein? So we learned about the different chromatographic techniques, right? So depending on the properties of a protein has different properties. Like it has a size, it has a shape, it has a particular charge, it has a hydrophobicity. So utilizing these different properties, we can utilize different chromatographic techniques and we can purify the protein. So what are the different techniques like size exclusion chromatography, ion exchange chromatography, hydrophobic interaction chromatography, all these and affinity chromatography, the four chromatographic techniques we learned. And then we learned about the how we can characterize the protein by certain spectroscopic methods, how we can sequence the protein. So characterization can be done either by sequencing, it can be done on the basis of the it can be done on the basis of the size of the protein as well. So there are different types of other than the spectroscopic techniques, there are different analytical techniques which can be used to determine the size of a protein, the shape of a protein. There are like light scattering experiments can be done. Then you can do analytical ultracentrifugation, so yeah, by which you can determine the hydrodynamic radius of a protein. So there are different techniques by there. Then there are if you want to understand, if you want to characterize the protein, then you can, if you have the knowledge about the secondary structure of the protein, then you can do a CD spectroscopy. You can do a UV visible spectroscopy. So all these different spectroscopic techniques, then about the information about the functional groups can be determined by by doing an IR spectroscopy. So these different spectroscopic techniques also, we learned about the spectroscopic techniques, and in the last module, we learned about the applications of the genetic engineering or application of biotechnology, how biotechnology is can be utilized for social welfare. Okay, so let's quickly revise these concepts today. Uh, okay, yeah, so hello everyone, I'm Akash Jyoti Pathak and I am one of the PMRFPs for this NPTEL course, Genetic Engineering. And this course is being run by Professor Vishal Trivedi of IIT Guwahati. So, what are the different topics that were covered? So, week 12 was mainly dedicated to revise all the concepts that that we have been learning in this entire course. Okay, so we started with the structure of prokaryotic and eukaryotic cell, and then we moved on to understand the uh, cell metabolism, right? So in cell metabolism, what are the pathways we learned? We mainly learned about the glycolysis, Krebs cycle, and the ETS, electron transport system, that is the oxidative phosphorylation by which the reducing equivalents NADH can be again be oxidized to give us the NAD plus, and the cycle can continue. Okay, which will or the NAD plus which will be produced will again be utilized in the glycolysis and the Krebs cycle pathway. Okay, for the and also ATP will be generated. Okay, so then after covering the cell metabolism, also we learned about the anaerobic pathway or anaerobic oxidation. Okay. Then after cell metabolism, we learned about the different enzymes which are involved in genetic engineering or enzymes which are involved in molecular cloning. Okay. Enzymes we so starting from the ligase, the restriction enzymes, the alkaline phosphatase, then clino enzyme, um, then T4. Okay, so T4 DNA ligase. Um, okay, so these are the then exonuclease. What are exonucleases? So all these different enzymes we learned. Then we also learned about how we can identify a fragment of gene. So if a genomic library, cDNA library, if the DNA sequence is known, if it is not known, then we can PCR amplify that particular gene product, and then we can clone it inside a vector. Okay, so what are the different vectors? So there are two types of vectors. One is the cloning vector and another is the expression vector. So how does an expression vector differ from a cloning vector? So both these vectors will have a origin of replication. They will have multiple cloning sites. Expression vector will also have multiple cloning sites. Both these vectors will have selectable markers. But the difference between an expression vector and a cloning vector is that an expression vector will have a promoter sequence. Okay, it will have a promoter sequence that will allow the expression of the gene. So that is, it will have a promoter sequence. It will have the gene sequence and it will also have the terminator sequence. Okay. So if these sequences are not present, then the gene will not be transcribed, isn't it? Because it is at the promoter site, your RNA polymerase will bind and it will start transcription of the gene. And also this promoter sequence, there will also be a ribosome binding site post the promoter sequence so that with the mRNA that is synthesized, that should have a ribosome binding site. That is 5' end. Okay, at the 5' end, there should be a ribosome binding site so that the ribosome, after the mRNA is transcribed, the ribosome can bind to the mRNA and then translate the protein, translate the mRNA to produce protein. Okay, or the mRNA can be translated to produce protein. So all these are ribosome binding sequence, the promoter sequence, all these sequences must be present in an expression vector, but while these sequences are absent in in a cloning vector. Okay, so this is the major difference between a cloning vector and an expression vector. Then, other than this, what after this we covered, we learned about the different ways we can screening of the recombinant host or the host which has taken our recombinant plasmid. So what are the different methods by which we can screen for a recombinant host or a transformed host from a non-transformed host? Okay. Or so a not only the host must be transformed, but the host must be transformed with a recombinant plasmid. Okay, so that is a recombinant host. So after that, we learned about the expression of the protein, how we can express the protein, expression of protein. Mostly it is done by IPTG induction in case of bacteria. So expression of the protein and then purification of the protein. So after purification, so what are the different techniques which are involved in purification? Those are the chromatographic techniques. So what are the different chromatographic techniques that we learned? ACC-IEX, affinity chromatography, and hydrophobic interaction chromatography. Okay. Then after the purification, then we learned about the downstream method. So how we can characterize the product. Okay. So we have purified the protein, but we have to perform some characterization experiments so that we can be sure that yes, the protein that we have purified, it is having that all the characters that is expected of the protein. Okay. Or whether your protein is folded, what is the oligomeric status of the protein? Does it exist as a monomer? Does it exist as an oligomer or a dimer? So all these are come under the characterization technique. So we use different biophysical or bioanalytical tools for characterizing the proteins, mainly how we learned about the different spectroscopic techniques. Okay. Spectroscopy. In spectroscopy, we learned mainly about the UV visible spectroscopy and the IR spectroscopy. UV visible, IR spectroscopy, and the mass spectroscopy also. How we can utilize mass spectroscopy for determining the size of our protein as well as determining the amino acid sequence of our protein. Okay. And the last topic that we covered is the applications of biotechnology. So In applications, we learned about the hybridoma technology, the gene therapy, how for synthesis of vaccine, and how biotechnology is being utilized for generation of these genetically modified crops or high yielding varieties of crops, right? And then pisciculture or fish, how in culture of fish, in culture of in agriculture, all these these different applications we learned about. So this is a summary of all the top, all summary of this entire course. What all were we have been discussing throughout the entire course. So we will go one by one. So let's quickly discuss the have a quick recap of all starting from module 1 and till module 11. We will try to cover the important points. Okay. So I'll not go into these different these because in 2 hours we cannot cover these because and we have already discussed these different aspects in much detail. So I'll try to revise the initial modules, okay, because it has been a long time till I'll try to cover the modules till module 6 or 7. And these techniques we have discussed recently, so you can go through your assignment questions. You can go through the lecture videos once again, and you can also go through this tutorial session, the concepts that we have discussed, and prepare for the exam. Okay, so prokaryotic cell, we know that prokaryotic cell is what is a prokaryotic cell? We know it is a primitive type of cell which lacks a defined membrane-bound organelles and it lacks a defined nucleus. Okay, it does not have a nucleus. Something that is present is called the nucleoid. Okay, it is a structure which is lacking the nuclear membrane and and the DNA that is there. It is what the DNA. The DNA is naked, isn't it? It is not wrapped around by histone proteins. Okay, it is naked and it is a single DNA or a single made of a single chromosome. Okay, single chromosome constitutes the DNA, and the DNA is freely present within the cytosol. It is not bound by a nuclear membrane. Okay, so there is no compartmentalization. So in prokaryotic cell, we learned about the two types of prokaryotic cell. One is so in prokaryotic cell, mainly the best example of a prokaryotic cell is that of a bacteria. So we have two types of bacteria. One is a gram-positive bacteria and gram-negative bacteria. How does a gram-positive bacteria differ from a gram-negative bacteria? So a gram-positive bacteria, if you see the structure, that a gram-positive bacteria has a very thick peptidoglycan layer. So you must remember that the bacteria cell wall is made up of peptidoglycan, and the peptidoglycan layer is different in a gram-positive and gram-negative bacteria. In a gram-positive bacteria, it is much thicker compared to a gram-positive bacteria. Okay, and in gram-negative bacteria, you can see that this layer, this peptidoglycan layer is thinner, and other than this peptidoglycan layer, we also have a gram-negative bacteria also have a outer membrane. Okay, it also has a outer membrane. So okay, so sorry for the confusion. So this is actually gram-negative cell wall. This is a gram-positive cell wall. So you can see this yellow colored cartoon representation. This is the peptidoglycan layer, and you can see this peptidoglycan layer is much thicker in a gram-positive cell wall, while this is much thinner. Is compared it is thin in gram-negative cell wall, and in gram-negative you also have an outer membrane. Okay, this outer membrane is lacking in a gram-positive cell wall, and other than this, the periplasmic space, because of the thin peptidoglycan layer, the periplasmic space is also thick compared to that in a gram positive cell wall. Okay, so these are the main, and the peptidoglycan layer is actually a polysaccharide. It is a It is a heteropolysaccharide which is composed of the repeating units of N-acetyl-glucosamine and N-acetyl-muramic acid. Okay, NAG and NAM units, alternating NAG and NAM units, and these NAG and NAM units, these are basically sugar moieties. Sugar moieties are they are linked by the beta-1,4 glycosidic linkage. To remember this, that this N-acetylglucosamine and N-acetylmuramic acid remains are linked by the beta-1,4 linkage, and also you can see that this these monomers are cross-linked. Okay, they are cross-linked, and this cross-linking is done by this peptide, this tetrapeptide, which is composed of L-alanine, D-glutamic acid, L-lysine, and D-alanine. Okay, so this you can see this cross-linking between one layer of the peptidoglycan and the low other layer of the peptidoglycan. So these blue colored lines, these are actually formed by the this tetrapeptide. Okay, so this much information is enough. Can remember these points. And what is gram staining? You know that it is a technique by which we can differentiate a gram-positive bacteria from a gram-negative bacteria. And gram-positive means what? When a gram staining will be done, this gram-positive bacteria will take up the stain. Okay, and the gram-negative bacteria will not take up the stain. Okay, or so so. So basically, it is a technique which was developed by the Danish scientist Hans Christian Gram, and the technique differentiates bacterial stains based on their cell wall composition, especially the thickness of peptidoglycan layer. Okay, so the differentiation between a gram positive and gram negative is on the basis of their mainly on the basis of their peptidoglycan layer. And what is done is, yes, what we do is we stain the bacteria with this dye crystal violet. Okay, and this crystal violet stain is taken up by the gram positive bacteria. While what a gram negative bacteria, what happens is that in the washing step or in this decolorization step, this crystal violet stain is washed off from the gram negative bacteria. It cannot retain the stain. Okay, so during a washing step with non-polar solvents such as alcohol or acetone, the gram-negative bacteria leaves the blue stain. Okay, it cannot retain the crystal violet dye due to the thin peptidoglycan layer, whereas a gram-positive bacteria, due to the thick peptidoglycan layer, it retains the dye, and as a result, this gram-positive bacteria appears stains blue, while this gram-negative bacteria has to be counter-stained with safranin because it loses the stain, and as a result, the gram-negative bacteria appears pink. Okay. So example of a gram-positive and a gram-negative bacteria. The example of a gram-positive bacteria is Staphylococcus aureus. Aureus, and a gram-negative bacteria is E. coli. Okay. So remember that the cell wall of bacteria is made up of peptidoglycan, and it is this peptidoglycan layer that differentiates a gram-positive bacteria from a gram-negative bacteria. The peptidoglycan layer is thick in gram-positive bacteria. It is thin in gram-negative bacteria. And the gram-negative bacteria also has a outer membrane. So this outer membrane has this lipotecoic acid and these polysaccharides, which other polysaccharide, which makes it, which provides the antigenic or immunogenic property to the bacteria. Okay, as a result of which it is able to cause infection. Okay. So these are, and the cross-linking is by this tetrapeptide, and the polypeptidoglycan is a polymer of two sugar units. One is NAG and another is NAP. And this NAG and NAP is linked by the beta-1,4 glycosidic linkage. Okay. So yeah, these are the few of the questions which are asked in your assignment. So remember these things, that the N-acetylglucosamine, N-acetylmuramic acid, the peptidoglycan, the linkage is what? It is beta-1,4 linkage. And the form of a bacterial genome. So remember, the bacterial genome is a single chromosome. It is a single chromosome. Eukaryotic genome has multiple chromosomes, but bacterial genome has a single chromosome, and it is usually circular. Okay. Now, how is an eukaryotic cell different? So in eukaryotic cell, you have a plant cell and an animal cell. That is, we have been reading about this animal differentiations between an animal cell and a plant cell. So plant cell has a cell wall, animal cell lacks a cell wall. Plant cells have this chloroplast. Animal cells will not have chloroplast. Um, then what are the? It has a large vacuole. Animal cells have a smaller vacuole. Then other than this, what are the other differences? So these are some of the differences between a plant cell and an animal cell. And how can we isolate or fractionate the eukaryotic cell? So what is meant by fractionation of eukaryotic cell? If we want to isolate the different organelles which is present in a eukaryotic cell, how can we do that? So we want to isolate the mitochondria, the ribosome, the nucleus, the endoplasmic reticulum separately. We want to fractionate it in we want the nucleus to be separate. We want to perform certain experiments on the chromatin, say for example. So we want to isolate the nucleus from the other components of the cell. So how can we do that? So one technique is the differential centrifugation, which we had covered. So remember, this technique, differential centrifugation, it is a technique by which we can separate the components of the cell on the basis of their size and density. Okay, so different components of a cell. What do we mean by different components of a cell? If you want to separate the ribosome from the nucleus, if you want to isolate the nucleus from the ribosome or the endoplasmic reticulum from the cell, if you want to separate the different components of the cell, how can we do that? So basically what we are doing, we are first in this technique, what we are doing, first we homogenize the cell. Homogenize the cell by physical or mechanical disruption or physical, mechanical, chemical, or enzymatic disruption. So first we have to disrupt the cell membrane. Okay, so that the disruption can be done by these different methods, by a physical method, mechanical method, chemical. We learned about these different methods, isn't it? So sonication, then bead mill, then French press, and enzymatic. We have this lysozyme treatment, chitinase treatment. Okay, so these are the different methods. First, we have to rupture the cell or the cell wall if it is a bacteria, and then what we do, we centrifuge it at different velocities or at different RPM for different time. So this is a homogenized cell. Homogenized cell. So this figure taken from the book Cooper, and so here the steps have been explained very systematically. So what happens is when this homogenized cell is first centrifuged at 800 G, what is the pellet that you get? So at 800 G for 10 minutes, when you centrifuge your cell suspension, the first pellet that is formed is that of the nucleus, because nucleus is out of all these organelles, the nuclear has a uh, high molecular weight, or the sedimentation coefficient of nuclei is higher compared to the other organelles. So at 800 G, at this small RCF, the nuclei get sedimented. After that, we collect the supernatant, and this supernatant is again undergoes a second round of centrifugation that is done at 15,000 G for 10 minutes. So when it is this supernatant is been at 15,000 G for 10 minutes, then uh, what component of the cell gets is sedimented? It is the ribosomes, or not ribosome. It is the mitochondria, lysosome, and peroxisome. Okay, and after that, again, third spin at 100,000 G or 1 lakh G for 60 minutes results in a sedimentation of the plasma membrane and the endoplasmic reticulum. And after that, a 200,000 G spin for 3 hours gives us the ribosome. So ribosomes are having the least sedimentation coefficient, or least molecular weight of the size. They are much lighter. So you need to spin it at a higher velocity and or at higher RPM and higher and a longer time for sedimenting the ribosome. So remember the order. So the first component of the cell that you get is the nuclei. So nuclei you get at when you spin it at 800 G, followed by what? Followed by that, when you spin it at 15,000 G, then you get the lysosomes, the peroxisome, and the mitochondria. Okay, so remember the order. So these type of questions can come in your exam. Mitochondria and peroxisome. So at which RPM do you get these components? At 15,000 G. Okay, at 15,000 G. Then after that, we get the we get the endoplasmic reticulum. So fragments is a, so fragments of the endoplasmic reticulum are sometimes called the microsomes as well. Okay, so whenever this endoplasmic reticulum frag, uh, is fragmented, it forms very small vesicle-like structures. So those are called the microsomes. Okay, so fragments of this plasma membrane and fragments of this endoplasmic reticulum. And then you get the ribosome. So this you get at 100,000 G, 1 lakh G, and this you get at 200,000 G. So, okay. So remember the order. Now answer this question. So the following organelle that will pellet down at 15,000 G for 15 minutes. So just now we discussed, so at 800 G, your nuclei gets pelleted down, or nuclei is pellet down. Then at 15,000 G, what is pellet down? Or what gets sedimented? It is your mitochondria, peroxisome, and lysosome. So answer is option B. Okay. So remember then, so 800 G followed by 15,000 G, then 100,000 G, and then 200,000 G. Okay. Okay, so the structural component that is shared by both plant and bacteria cells, but not by animal cells. So I'll read the options. It is nucleus, chloroplast. So it is both are eukaryotic. So plant is a prokaryotic. Plant is a eukaryotic cell. Bacterial cell is a prokaryotic cell. Okay, so what are the how? So this is a eukaryotic cell, and so what? How does a eukaryotic cell differ from a prokaryotic cell? So the structural component they have asked. Okay, so that is why the answer is option C, cell wall. The structural component is based on the structure, just the structure, not the internal components. So both eukaryotes and prokaryotes both will have nucleus, but one is having a primitive type of nucleus and another is having a true nucleus. Okay, so so option E is not correct, but the answer is option C. So this is an easy question. Animal cells contain more mitochondria compared to plant cells. These were asked in your assignments actually. So why do an animal cell? Because animal cells have to perform the function of locomotion or movement. So as a result, they require more energy compared to a plant cell. So which is that? More energy will be provided by which organelle? Mitochondria, because in mitochondria, it is in mitochondria where your ATP is getting synthesized to oxidative phosphorylation. That electron transport chain that I was talking about, where your NADH and the proton gradient, that when that will be broken, your ATP will be generated. So answer is option A, because animal cells are more energetically active. That is why they require more energy and hence they will have more mitochondria in their cell. Now plasma membrane. These points are clear, isn't it? Okay. So plasma membrane is the outermost covering of a cell, isn't it? So what are the so we know that plasma membrane is composed of the lipid bilayer. So lipid bilayer will they are mainly the phospholipids, isn't it? So the phospholipid, they have a head group, they have tails. The head group is formed by the mainly by the phosphate group, which makes it hydrophilic, while the tails are formed by the fatty acid chains, which makes it hydrophobic. Okay, so basically it is an amphiphilic molecule because it has both hydrophobic and hydrophilic groups. And other than the plasma membrane is involved in several functions, and these several functions are conferred by the several kinds of proteins which are present on its surface. They have receptors which allows the plasma membrane to recognize different substrate, or they have channel proteins which allows a molecule to enter the cell. They have ATP or pumps that will allow the cell to maintain a osmotic gradient, or to maintain a particular salt concentration within the cell. So you can see here that this plasma membrane has different types of proteins on its surface, integral protein and peripheral protein. So integral protein will span throughout the phospholipid bilayer. Then there are these glycoprotein that will allow the cell to recognize, or cell recognition. How will one cell differ from the other, or like the glyco. So then there are receptors which will allow the cell to bind to a ligand. Okay. So for cell communication, your cell receptors are important. There will be transporters. So the different types of proteins can be present. There can be transport proteins like your sodium potassium transporters. There can be receptors like LDL receptor that will allow the low density lipoprotein to be like your liver cell. They will have LDL receptors. Then you have enzymes like adenylate cyclase that will convert your ATP to cyclic AMP. Then there are signal transducers like insulin receptors. Signal transducers means it will convert the signal. So mainly these signal transducers are involved in cell communication. So the information that your insulin has to be communicated within the cell to transcribe a particular gene, or start the activation of a gene, or to synthesize a particular protein, that yes, my insulin is being released, and now the glucose which is present in the blood has to be absorbed. Okay, your insulin function of insulin is what to utilize to lower the blood glucose level. So how are these functions getting coordinated within the cells? So those functions are being coordinated by the signal transducers. So the receptor protein. So the information which is present outside the cell. So insulin is being secreted by the pancreas. Now this insulin that the pancreas has secreted, that will go and bind to the hepatocytes. It will go and bind to your tissues. It will go bind to your fat cells, so that the blood glucose, or the glucose which is circulating in the blood, can be utilized. So that signal transfer from the outside environment to into from the outside to within the cell, those are being performed by these different signal transducer, the insulin receptor. There are different GPCRs. Then there are other kinds of receptors, and Other than this, the function of plasma membrane is protection because it is acting as a boundary, outermost boundary. So that is for providing protection to the cell. It is involved in communication. Communication is being mediated by the different receptors present on the protein, the different signal transducers present on the surface of the present on the plasma membrane. And then there are channel protein, there are pumps, which is selectively allowing the entry and exit of ions within the cell. And it is also providing recognition. Recognition is being provided by these glycoproteins. Okay. So the plasma membrane contributes to cellular recognition by synthesizing proteins, by transporting ions through recognition proteins that identify other cells. So answer is option C. This is a direct question. A particular protein can be overexpressed in one cell, or a surface marker proteins. Like when you read about immunology, there are different surface marker proteins that this natural killer cell have this. Say for example, has this particular CD. Whatever F9 or this protein is being overexpressed, and this is acting as a marker protein for the cell. That this is a natural killer cell. This is a macrophage cell. So these different recognition proteins are expressed on the surface, which differentiates one cell from the other cell. Okay. Now coming to cellular metabolism. So why cellular metabolism is required? Because you require energy, right? The cell needs energy to perform the different functions. So all the different reactions that is taking place in the cell, the replication, transcription, translation, right? All these different processes. The protein has to be synthesized. The DNA polymerase has to unwind the DNA, right? Or the helicase has to come and unwind the DNA. DNA polymerase has to bind to the DNA and start replicating the gene, the genome, or so all these different processes, they require energy. So how does the cell produce energy? So we are eating food, right? But the food is getting broken down into this glucose and amino acids, and your fats are getting broken down into glycerol, lipids. So all these are ultimately getting converted into glucose. Now this glucose, when it enters the cell, this glucose is broken down and energy is produced. So what is the pathway by which the glucose is broken down to generate energy? So that comes under cellular metabolism. So there are two types of metabolic pathway. One is the anabolism and another is catabolism. Anabolism means what? Anabolism means where the biosynthetic pathway. Biosynthetic pathway means something is being formed, while catabolism is your something is getting broken down. So for your catabolism will result in energy production, while your anabolism, when something is, so if you want to build something, then you have to put in effort, right? You need to spend energy. So that is your anabolism. Anabolism is biosynthetic pathway that will require energy, while catabolism means if you have to means catabolism means your is getting broken down, and that the energy that is trapped within the bonds, those will be released. So those energy gets is stored in the cell in the form of ATP. Okay, so that is catabolism. That will result in energy producing. So your glucose, your glycolysis, what it is doing, your glucose is getting broken down. So it is a, you can say it is a catabolic pathway, because your glucose is getting broken down into pyruvate, and so your metabolic pathway, we cannot say it is completely catabolic or anabolic, because there are many intermediates that are getting produced as well. So your even your Krebs cycle, it is an amphibolic pathway. Amphibolic pathway means a pathway where both catabolism and anabolism is involved. Because some products are getting synthesized, and these products are getting, yeah, are being utilized in other pathways, in your purine, pyrimidine, biosynthetic pathway, in synthesis of the sugar. So it is, um, in one sense, it is your glucose is broken down, or your pyruvate, or as converting to acetyl CoA, and acetyl CoA being converted to to produce carbon dioxide and energy, but at the same time, certain other molecules are also getting produced, which is utilized in other metabolic pathway, like your DNA synthesis pathway and your sugar pathway. Okay. So now coming to glycolysis. So glycolysis is the 10-step metabolic pathway, isn't it? So out of these 10 steps, there are a few steps which you need to remember. Those are the steps where your either ATP is getting utilized or your ATP is being formed or you're reducing equivalence like NADH is getting formed or NADH is getting converted to NAD plus. These are the pathways that you need to remember. So glucose in the first step, glucose converted to glucose 6 phosphate by this enzyme hexokinase. Now this is a substrate. So this is what your ATP, your the phosphate which is present on ATP is getting transferred to glucose, right? And you are getting glucose 6 phosphate. So ATP is utilized. So any power reaction where ATP is involved, the enzyme is named as kinase. So your kinase enzyme is transferring the phosphate group from the ATP to your glucose at the carbon 6 position. Then in the next step, this is a isomeric reaction. Glucose and fructose are isomers of each other. That is why this enzyme that is involved is called the phosphohexose isomerase. Where glucose 6 phosphate is converted to fructose 6 phosphate, then fructose 6 and phosphate is converted to fructose 1,6 bisphosphate. So this is the uh, energy requiring steps. So the first 5 steps, like glucose getting converted to glucose 6 phosphate, and then your fructose 6 phosphate getting converted to fructose 1,6 bisphosphate. These two steps require energy. So their ATP is required. Then in the next step, this fructose 1,6 bisphosphate is broken down into two uh, two molecules of 3 carbon compounds. So one is the glyceraldehyde 3-phosphate and another is the dihydroxyacetone phosphate. So aldolase is the enzyme which converts this fructose 1,6 bisphosphate into these two different 3 molecule compounds, this glyceraldehyde 3-phosphate and dihydroxyphosphate. So after this is broken down, then from this step, this is the payoff phase. Payoff phase of the energy yielding phase, or because after these steps your ATP is being found, while in the earlier steps your ATP is required. Okay, but here your ATP is formed. So glyceraldehyde 3-phosphate is first broken down into glyceraldehyde 1,3 bisphosphoglycerate, 1,3 bisphosphoglycerate, and this step results in the formation of one molecule of NADH. Okay. So these steps you remember, right? So 1,3, remember these steps. 1,3 bisphosphoglycerate. Here you get one molecule of NADH, and then when this phosphoenol this 1,3 bisphosphoglycerate getting converted to 3-phosphoglycerate. This is the substrate level phosphorylation, right? And here also you get one molecule of ATP, and last step, which is phosphoenol pyruvate being converted to pyruvate. So here in all you can see in all the steps where ATP to ATP or ATP to ATP, some phosphoryl group transfer of phosphoryl group is involved. Those the enzymes involved in such type of reactions are called kinases enzyme. And so what happens is that this acetone phosphate also gets converted to glyceraldehyde 3-phosphate. So this, as a result, you get 2 molecules of each of these, 2 molecules of NADH, 2 molecules of ATP, and 2 molecules of ATP, right? So this is a reversible. So this also that is dihydroxyacetone phosphate has also needs to be converted to glyceraldehyde phosphate. So this pathway actually takes place. Everything is after this step. Everything will be produced even one cycle will give you 2 molecules of each of these products. Okay. So if you do a glycolysis ATP balance sheet, so in steps 1 to 4, so this is your first step, this is your second step, this is your third step, and this is your fourth step. So in the first 4 steps, in your step 1 and in your step 3, your ATP molecule is getting utilized. So 2, so that is why -2, because 2 molecules of ATP are required. And then from starting from this fifth step, in your fifth step, where your glyceraldehyde 3-phosphate is getting, or this is actually the fifth step, and this is your sixth step. In the sixth step, where glyceraldehyde 3-phosphate is getting converted into 1,3-bisphosphoglycerate, that step gives you 1 molecule of NADH, and this actually takes place twice, because this dihydroxyacetone phosphate also gets converted to 3-phosphoglyceraldehyde, and that is why you get actually 2 molecules of NADH. And when this NADH molecule goes through this oxidative phosphorylation, 1 molecule of NADH gives you 3 molecules of ATP. That is why 2 NADH molecule will give you 6 molecules of ATP, right? And then there are 2 steps in the step number 7 and the step number 10. Step number 7, where 1,3-bisphosphoglycerate is getting converted into 3-phosphoglycerate, and your phosphoenol pyruvate getting converted into pyruvate. Those 2 steps will give you, each step will give you 2 molecules of ATP. So you will have 4 molecules of ATP. 4 molecules of ATP and 6 molecules of ATP from this NADH getting converted to NAD plus in the oxidative phosphorylation step. So total, if you add up all these and if you subtract the 2 ATP molecules that were required, so that gives you 8 ATP molecules. Okay. To glycolysis, where does glycolysis take place? Glycolysis take place in the cytoplasm. So all these enzymes, they are present in the cytoplasm. Okay. And in this step, glycolysis does not require oxygen. It can take place in absence of the oxygen. It is a universal pathway. All organisms, be it bacteria, be it yeast, be it any prokaryotic, any eukaryotic cell, uh, uh, all living organisms has this particular metabolic pathway taking place within them. So that is why it is a universal pathway, and it can take place in anaerobic condition, and then this glycolysis takes place in cytoplasm. Okay, so these are some of the few important points. So remember the steps. Like glucose to glucose 6-phosphate, your ATP will be required, and fructose 6-phosphate to fructose 1,6-bisphosphate. So the first step and the third step, your ATP is getting utilized, and which steps your ATP is getting produced. So your, yeah, 3-phosphoglycerate getting converted to, or 1,3-bisphosphoglycerate getting converted to 3-phosphoglycerate. You will get 2 molecules of ATP, because that will take place twice, and your phosphoenol pyruvate getting converted to pyruvate, that will again give you 2 molecules of ATP. So total 4 molecules of ATP and 6 molecules of ATP you get from the production of NADH. That is in the sixth step, where your glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. Okay. Another thing is that whenever you have this NADH being produced, so those the enzymes which are involved in these type of reactions, they are called as dehydrogenases. Dehydrogenase, because your hydrogen which is present in NADH, is your NADH is getting oxidized. And how is it getting oxidized? Your hydrogen is being removed. So removal of hydrogen is called oxidation. That is why it is a dehydrogenase. So total from glycolysis, you get 8 molecules of ATP. Remember this. Then what happens? Your pyruvate will get converted to acetyl-CoA, and your acetyl-CoA moves to the mitochondria. So all the steps of Krebs cycle takes place in your mitochondria, right? So in your mitochondria, what happens is this acetyl-CoA. Your acetyl-CoA and oxaloacetate will give you a, will combine and give you citrate. Citrate is a 6-carbon compound. It is tricarboxylic acid. So that is why the first product that is produced is called citrate, and that is why Krebs cycle is also known as TCA cycle, tricarboxylic acid cycle. Okay. Acetyl-CoA is a how many carbon compound? Your pyruvate is a 3 carbon compound, isn't it? Acetyl-CoA is it has 2 carbons, while your oxaloacetate has 4 carbons. That is why your 4 carbon plus 2 carbon gives you a 6 carbon compound, which is citrate. C3COO COA. So that is acetyl-CoA. Okay, then again you have to remember few steps of the Krebs cycle. So what are some of the important steps in the Krebs cycle? So remember the first step where your oxaloacetate uh, on reaction with this acetyl-CoA, you get citrate molecule. Okay, and there are actually two steps where carbon dioxide is getting or when the decarboxylation reaction is taking place. So basically you have two molecules of pyruvate. So this entire cycle will run twice. To for complete oxidation of your glucose, this cycle has to run twice because you will have two molecules of pyruvate from one molecule of glucose, and then everything will be multiplied by two after this from this step onwards. So you have two molecules of pyruvate. From there, you will get two molecules of acetyl-CoA, then two molecules of each of these intermediate products. Okay. So in one of these steps, so your in this step, isocitrate getting converted to oxalosuccinate. Here you get one molecule of NADH. And then when this oxalosuccinate to alpha ketoglutarate gives you, this is a decarboxylation reaction that takes place. And then when alpha ketoglutarate gets converted to succinyl-CoA, here you get another molecule of NADH. And then in this step, malate to oxaloacetate, you get again a third molecule of NADH. And from succinate to fumarate, you get another molecule of FADH2. So oxidation of NADH will give you 3 molecules of ATP. Okay, so basically you have 3 steps where your NADH is being formed. One is when your isocitrate is getting converted to oxalosuccinate, your alpha ketoglutarate getting converted to succinyl-CoA. Okay, and your malate is getting converted to oxaloacetate. So these are the 3 steps where NADH is produced. So that is why one molecule of NADH gives you 3 molecules of ATP. So 3 into 3 will get total 9 molecules of ATP. Right? And you have one step where FADH2 is being produced. So FADH2 oxidation gives you 2 molecules of ATP. Okay, so one FAD in one cycle, you are getting one FADH2 molecule. So one FADH2 molecule will give you two ATP molecules. So if we look at this Krebs cycle ATP balance sheet, so production of acetyl-CoA. So even that step where your pyruvate is getting converted to acetyl-CoA, that also results in synthesis of one NADH molecule. So one NADH molecule, so one NADH will give you 3 ATP. That is why 3 into 1, 3 ATP molecules you will get from production of acetyl-CoA, then generation of alpha ketoglutarate. Alpha ketoglutarate, so this step results in synthesis of one NADH molecule. So you will get 3 ATP molecules. Then your generation of succinyl-CoA, succinyl alpha ketoglutarate to succinyl-CoA. That will give you again another molecule of NADH. So one NADH molecule will give you 3 ATP. That is why 3 into 1. Then at this step, generation of ATP, succinyl-CoA to succinate. Here you will get one ATP molecule, generation of fumarate. One FADH2 gives you two ATP, that is why 2 into 1, 2. Then oxaloacetate, that is malate to oxaloacetate. They will get again another molecule of NADH. So that is why if you add up all these, all the, these ATP molecules, how many ATP molecules are you getting? You are getting 15 ATP molecules. Okay, 15 ATP molecules, and this cycle will take place twice because you have two molecules of pyruvate, so everything is multiplied by two. That gives you 30 molecules of ATP. And from a glycolysis, how many molecules of ATP did you get? 8 molecules of ATP. Okay. So remember a few of these steps. So remember the steps when which your ATP is being produced. So what are the steps? So your NADH is being produced in which steps? When your pyruvate is getting converted to acetyl-CoA. Pyruvate getting converted to acetyl-CoA. That will give you one molecule of NADH. Then what is the other step where your NADH is being produced? Your succinyl-CoA, when your alpha ketoglutarate is getting converted to succinyl-CoA. Succinyl-CoA, right? Then what is the next step where so FADH2 is being produced when? In this step, when your succinate is getting converted to fumarate. So that is the step where your FADH2 is produced. Okay, succinate getting converted to fumarate. Fumarate, then in the another step, where your malate is getting converted to oxaloacetate, where also you are getting one NADH2 molecule. Malate getting converted to oxaloacetate. Okay, so in this step you will get one NADH molecule. This will give you another NADH molecule. This will give you a FADH2 molecule. And this will also give you an NADH molecule. So one NADH will give you three molecules of ATP. So 1 into 3, 1 into 3, one FADH2 gives you two ATP. So 1 into 2, and this is 1 into 3. So if you add up these, then how many molecules of ATP are you getting? 3, 6, 9, 11. 11 ATP molecule. Okay, and then if you consider that also, your this step, isolate. Then other than this, you have one of the steps where your GTP is directly where your substrate level phosphorylation is taking place. So substrate level phosphorylation is when your succinyl-CoA is getting converted to succinate. Okay, succinyl-CoA getting converted to succinate. That will give you one GTP molecule. That will give you one GTP molecule, and your generation of fumarate as well. Your alpha ketoglutarate to succinyl-CoA, that will give you NADH, and also your synthesis of alpha ketoglutarate. That is from citrate to alpha ketoglutarate in this step, where your citrate is getting converted to alpha ketoglutarate, that is also yielding one NADH molecule. Okay, you're basically your citrate getting converted to isocitrate, isocitrate getting converted to alpha ketoglutarate. That will also give you one NADH molecule. Okay, so from one NADH, I actually will get 3 ATP. So total 11, 12, 13, 14. Total you will have 15 ATP molecules. Okay, and this cycle will take place twice. So that is why this 15 has to be multiplied by 2 and you will get 30 molecules of ATP. Also, these questions during glycolysis, 1,6-carbon atom molecule split into 2, 3-carbon atom molecules. So which is the step where they are talking about in this question? Which is the step? Anyone? So do you remember the step where your fructose 6-phosphate is broken down into one molecule of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So that is the step where your 6-molecule carbon is getting broken down into 2, 3-carbon atom molecules. So that in that step was catalyzed by which enzyme? Aldolase. Okay, so remember that step. So that is an important step. So your aldolase is catalyzing the conversion of fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This step, fructose 1,6-bisphosphate being converted to into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Okay, right. Then coming to this question, enzymes required for glycolysis are presented. So glycolysis takes place in the cytosol. Krebs cycle takes place in the mitochondria. Electron transport chain, oxidative phosphorylation, all of these are taking place in the mitochondria. So if you want to say more specifically, then mitochondrial matrix is the place where Krebs cycle is taking place, and the inner mitochondrial membrane is the site where your oxidative phosphorylation or your electron transport chain is taking place. Okay, so the enzymes required for glycolysis are present in mitochondrial matrix. You know, the enzyme required for glycolysis are present in cytosol. For Krebs cycle, it is mitochondrial matrix. Okay, which of the enzyme catalyzed step a GTP molecule produced? So, which is the enzyme which catalyzes the production of GTP molecule in the Krebs cycle? So, your GTP molecule is produced in the Krebs cycle or in the TCA cycle. So, so what is that step? So, you do not have to remember all the enzymes, but all you have to understand that whenever the phosphoryl transfer reaction is taking place, the enzymes that are involved will have this kinase enzyme. Okay, so that is why our answer is option C. Even if you do not remember the name of the enzyme, but something in this question, they are talking about the phosphoryl transfer reaction. So whenever a phosphoryl transfer reaction is involved in any enzymatic reaction, the enzyme that will catalyze this reaction is a kinase enzyme. Okay, and whenever there is something NADH being formed, right, from NAD plus, there will be or NADH getting oxidized to NAD plus. So those reactions will be catalyzed by a dehydrogenase enzyme. Okay, so what is the reaction that takes place when GTP is produced? So when your succinyl CoA getting converted to succinate, so that step not GTP is not formed. That is the step where your NADH is formed. But your succinate getting converted to your succinyl CoA getting converted to succinate. That is your step where your GTP is synthesized. So in this step, succinyl CoA getting converted to succinate. Okay. So succinyl CoA getting converted to succinate. So this reaction is catalyzed by succinate thiokinase. Succinate thiokinase. Succinate thiokinase will form GTP from GDP. Okay. And how is succinyl-CoA in this step? A succinyl-CoA is being formed from alpha-ketoglutarate. Alpha-ketoglutarate. So this step, you will get a NADH, right? And when your succinate is getting converted to fumarate, that is the step where your FADH2 will be produced. And when you are getting alpha ketoglutarate, that step is also, you will get alpha ketoglutarate from citrate or citrate, isocitrate, then cis aconitate. So that step will also result in synthesis of one NADH molecule. And then fumarate to malate, malate to oxaloacetate. So here you will get another NADH molecule, malate to oxaloacetate. So in subsequent steps, how many? So remember this answer is option C. In subsequent steps, how many TCA cycles would be needed if one glucose molecule undergoes glycolysis? So basically, one molecule glucose when it is undergoing glycolysis will give you two molecules of pyruvate. So that is why for uh, that is why two cycles of Krebs cycle is required for complete oxidation of one molecule of glucose. Right? The answer is option D. So one glucose will give you two pyruvate. So these both the pyruvates will have to go through this Krebs cycle. So that is why this cycle will take place twice. Answer is option D. Then complete oxidation of one glucose molecule yields how many ATP molecules? So from glycolysis, how many ATP molecules did you get? From glycolysis, you were getting 8 molecules of ATP. Okay, and from Krebs cycle, you were getting 30 molecules of ATP. Do you remember the steps? That is why the total number of ATP molecules is 38. So what are the steps in glycolysis? What are the steps in Krebs cycle? So remember those steps we covered just now. So glycolysis, the steps where your ATP is getting produced is one is when your 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. And the second step is where your phosphoenol pyruvate is getting converted into pyruvate. Those are the two steps where ATP is produced, and another 4 ATP molecules you are getting from the oxidative phosphorylation of NADH. And this NADH is produced when your glyceraldehyde 3-phosphate is converted into 3-phosphoglycerate. Isn't it? This step, glyceraldehyde 3-phosphate is getting converted into 1,3-bisphosphoglycerate. Okay, so answer is 30. Now, other than this, there is anaerobic oxidation. So in anaerobic oxidation, what happens when you're in heavily exercising body muscles, or when this glucose or this oxygen is lacking, uh, oxygen lack of oxygen condition, what happens? Your this Krebs cycle will not take place. Okay, because for Krebs cycle to run, you require oxygen. Okay, so then in those conditions, only glycolysis will take place. So that is what has been shown in this figure. So in the presence of oxygen, this TCA cycle or this Krebs cycle will take place. While in the absence of oxygen, your anaerobic oxidation will take place. So what happens in anaerobic oxidation? The anaerobic oxidation means only your glycolysis is running. So now for glycolysis to run, there also you require NAD plus. So who will give the NAD plus? So in that case, your pyruvate is converted into lactate, or it is converted into ethanol. So when your pyruvate is converted into your lactate, your NADH is regenerated, or your NAD plus is regenerated. NAD plus is regenerated. Okay. So in the absence of oxidation, oxygen, there can be two pathways that can take place. One is where your lactate dehydrogenase pathway, and another is the alcohol dehydrogenase pathway. So in lactate dehydrogenase pathway, your pyruvate is converted into lactate, and in your alcohol dehydrogenase pathway, your pyruvate is first converted into acetaldehyde, and then acetaldehyde is converted into ethanol. Okay, so this ethanol, synthesis of ethanol has some commercial benefit because this is this pathway which is utilized in the brewing industry for synthesis of ethanol. So that is the significance of anaerobic oxidation. So it has industrial relevance because your anaerobic oxidation is resulting in fermentation of the dough. Fermentation of the dough because your carbon dioxide that is released. The carbon dioxide that is released makes the dough fluffy and soft. While this lactic, also the lactic acid that is produced is useful for making curd, and the ethanol that is produced that is useful in brewing industry for different types of beverages. Different types of beverages. Okay. Now, under anaerobic conditions, pyruvate is converted to lactate by lactate dehydrogenase enzyme and requires gas as energy source. So, what is the answer? Pyruvate getting converted to lactate by lactate dehydrogenase enzyme that requires. So basically, in that step, your NADH is again getting oxidized to NAD plus. So answer is NADH, isn't it? NADH. So in both of these steps, either your pyruvate getting converted to lactate or your pyruvate getting converted to acetaldehyde, followed by acetaldehyde being converted to ethanol. So this is catalyzed by lactate dehydrogenase. And this is catalyzed by alcohol dehydrogenase. So in both of these steps, what is happening? The NADH that is produced, this NADH is again getting oxidized to NAD plus. Okay. Here also the NADH is getting converted to NAD plus. This thing. So answer is NADH. So other than this, your growth medium. For the growth of your bacteria, you require a growth medium. So your growth medium should have all the essential vitamins, essential sources of carbon, hydrogen, oxygen, nitrogen. So protein, carbohydrate, lipid, DNA, RNA for the synthesis of all these biomolecules, macromolecules, amino acid, sugar. You need these all these. These are the building blocks. Are what they are? CHONS: carbon, hydrogen, oxygen, nitrogen, sulfur. For the energy, you require these carbon, hydrogen, oxygen. So you have to when you are growing the bacteria uh, in a media, your culture media should have all these essential components. It should have protein, amino acid, it should have carbohydrates, lipids, all these different. And when a bacteria goes, it has three phases: lag phase, log phase, and a stationary phase. Lag phase, log phase, stationary phase, and a death phase. Okay, so these things are were discussed, isn't it? So correct order of various stages involved in bacterial growth curve. It is what? First, there will be a log phase. Why is there a log phase? Because it is at this stage where your bacteria gets acclimatized to the environment and it starts synthesis of different enzymes, the metabolic pathways. that are required for the growth and division of the bacteria, all the different proteins, the RNA molecules, the synthesis of these molecules will start. That is why there will be a log phase. Then this log phase will be followed lag phase. Lag phase will be followed by the log phase. Okay, where the bacteria will divide exponentially. So that log phase is also called the exponential phase. Then after a certain period of growth, what will happen? The nutrients will get depleted. So when the nutrient starts getting depleted, the bacteria will reach a stationary phase where the rate of growth and the rate of death becomes equal. So that is a equilibrium is reached. So that stage is called the stationary phase where you cannot see any observable growth in the bacteria. So that is your stationary phase. And then even after the exponential, the stationary phase, when you grow the bacteria, then what happens is that the environment or the bacteria starts secreting toxic materials and the nutrients gets depleted, and at this stage the bacteria starts getting an acidic environment starts to build up in the culture, and your bacteria moves to a death phase. So now answer this question. So during the exponential phase, the what happens is the exponential growth curve is n equal to n naught 2 to the power n, isn't it? So that is the equation that is followed. So any exponential stage number of bacteria. So n is basically the number of bacteria that will be produced after a particular time t. And n naught is the initial number of bacteria, and small n. What is small n? Small n is the number of generations, number of divisions, or number of generations. So in this question, your n naught is what? So during this exponential phase, we follow this equation n equal to n naught 2 to the power n. So initial number of bacteria is 10,000. Okay, and then what is the number of generation? 5. So that is why the answer is 32. Okay, 1, 2, 3, 4. 1, 2, 3, 4. So it is 32 lakh 20,000. So this is the number of bacteria that will be formed after 5 generations. You can also be asked to calculate the n. So the how many bacteria? N is the number of generations, the doubling time of the bacteria. So those type of questions were also discussed. You can go through that particular lecture video. Okay. So basically, T is N into T half. T half. So if you know the time for which the bacteria was cultured, and if you know the doubling time of the bacteria, then you can calculate the number of generations. T by T half. T half is the doubling time, and T is the duration for which you have cultured the bacteria. You have cultured it for say 72 hours, and the doubling time is 20 minutes. Then accordingly you can calculate the number of generations, or the number of generations of the bacteria. So during which growth phase bacteria synthesize important enzymes or coenzymes required for cell division. So it is during the log phase. So during the log phase is a log phase is observed when the bacteria will start to multiply, and it will start to synthesize the essential enzymes, the RNA, the protein that is required for the growth and division of the bacteria. So the answer is log phase. Sorry, lag phase. Lag phase. This phase. So this is the lag phase where the bacteria will synthesize the important enzymes and coenzymes which is required for the cell division. Okay. Now different approaches for isolation of gene of interest. we learned about this technique, this part as well. So if the gene sequence is not known, then what do we do? Do we do a PCR amplification? So if it is not known, then from the genomic sequences, we amplify a particular section of the genome, and then once the amplified product is obtained, then we clone that amplified product, and then we screen that the screen for the desirable protein or the desirable gene of interest. Okay, but if the gene of interest is known, then we can either go for the genomic library or we can go for the cDNA library. So the genomic library and the cDNA library will have the copy of all the genes which are present in that particular host cell. So there can be bacterial genome library, which will have all the library of all the genes which are present in bacteria, or there can be a cDNA library where only those gene genetic sequences will be present, where which will code for a protein. Okay, then we can screen and then we can do the clone. We can clone that particular gene of interest inside inside the vector and express our protein. Okay, so how do you construct a genomic library? So for construction of a genomic library, first of all, the first step that is required is to isolate the genome. So we will see this slide first. So for the isolation of the genome, what are the steps involved? So for isolation of the genome, you will first need to lyse the cell. So how do you lyse the cell? So you treat it with detergent containing lysis buffer, and then the cells are incubated with the digestion buffer containing protease K SDS. So when you treat after you have lysed the cell, then you treat the cell lysate with this protease K and SDS. So when you treat it with protease K, what happens? The histone proteins with which the DNA is associated, they get cleaved or they are digested. So now you have the free DNA, and your and also your SDS will help to lyse the cell membrane as well, of the membrane proteins, and all these, they will be solubilized. So after that, what is done is basically you do a phenol isoamyl alcohol treatment or a phenol chloroform treatment, phenol chloroform isoamyl alcohol. So three components are there. So when this fraction is treated with this particular reagent, then what phenol does is that it phenol precipitates the protein, or it's the protein fraction which is present. They it is will move to the organic phase basically, and your aqueous phase will have the DNA. Okay, so now the aqueous phase is taken out, and the aqueous phase is precipitated by treating it with 100% ethanol. Okay, so when you precipitate with 100% ethanol, so what happens is your is the DNA which is present in your aqueous fraction that gets precipitated, and then you can recover this precipitate. You can air dry it, and then you can resuspend it in water, or in particular 3 CDTA buffer, and then that fraction will have your genomic, or you can again give another 70% ethanol wash, and then that pure fraction that you got, you can run a 0.8% agarose gel and analyze the purity of your genomic DNA. Okay, is it clear? So remember the step. First step is lysis of cell. Second step is enzymatic digestion. Okay. How do you do an enzymatic digestion? So you treat it with protease K that will digest all your proteins, and then you, after enzymatic digestion, you treat it with absolute alcohol. So absolute alcohol will precipitate your DNA, and along with it, proteins will also get precipitated. So you purify it with chloroform phenol. So your phenol will cause the protein to move into the organic phase, but your DNA remain in the aqueous phase. Okay, so once you have the genomic DNA, then you can restriction digest the genomic DNA with a commonly used restriction enzyme, which is EcoR1. Okay, you treat it with EcoR1. So there are multiple at multiple sites in the genome, their genome will get fragmented. Okay, so that will give you large genome fragments, and then you can clone these genomic fragments in vector and then transform it in a host cell and prepare a genomic library. So you isolated the genomic DNA by this treatment with isoamyl alcohol and phenol chloroform method. And then you restriction digest the genomic DNA with EcoR1. So this is the most commonly used because there will be multiple, this EcoR1 has a particular restriction sequence, and this restriction sequence will be present at multiple sites within the genome. So it will randomly or it will give you multiple DNA fragments. So once you have the DNA fragments, you clone it in a vector, and then you transform this cloned vector within a host cell. Okay. And then you create a GS. So that all the host cells having the host cells having these different genomic fragments will is actually called the genomic library. On another method is by the cDNA library. So how do you get a cDNA library? So basically, your cDNA library will contain only the information of the cDNA library will only contain the exon regions of the genome, while your genomic library will contain both the exon and intron regions. Okay, only the exon regions are those regions which will code for protein. So basically, how do you create a cDNA library? You isolate the RNA, mainly the mRNA. So you have to isolate specifically the mRNA, not the rRNA and the tRNA. Then from the mRNA, you will do a reverse transcription, and then you will get the cDNA. Now this cDNA will be cloned in a vector, and then then it will be transformed to a host cell. So that process of creating the cDNA cloned vector and transforming it into a host cell, that will give you a cDNA library. So there are what are the different steps? First, you have to isolate the mRNA. After isolate the mRNA, you have to purify the mRNA by passing it through oligo-DT column, and then you have to do a restriction reverse transcription PCR by treating it with reverse transcriptase enzyme. So mRNA will give you cDNA, and then you clone the cDNA in the vector system and then transform it in suitable host cell. You see this, you have isolated the mRNA, then you have done a reverse transcriptase by treating it with reverse transcriptase, you get DNA. Then this DNA is cloned in a bacterial plasmid. Then once you have cloned it in bacterial plasmid, you transform this cloned bacterial plasmid within the bacterial host cell. Then you isolate the plasmid because you do not know, right, whether your plasmid has this insert within it. So you have to isolate the plasmid, you have to restriction digest it or PCR amplify it and then sequence it. Only after sequencing you will be confirmed that yes, my plasmid or which of these host cells have the recombinant plasmid and which do not have the recombinant plasmid. Okay. So the correct order of various reagents used in different steps during genomic DNA isolation is what? So first, so in this step, so if you remember the steps, so first step is what you treat it with. So first of all, you have to resuspend the bacteria basically. So you have to treat it with tris HCl buffer, tris HCl, EDTA and glucose. So that you have to treat it with, and then after resuspension, you need to treat it with proteinase K, SDS. So your proteinase K and SDS will lyse the membrane, and it will also degrade the histone proteins with which your DNA are associated. And then you need to treat it with phenol chloroform mixture. So when you treat it with phenol chloroform mixture, your chloroform will make the protein move into the organic phase while your DNA will remain in the aqueous phase. Then you isolate that aqueous phase, you take out the aqueous phase, and then you precipitate it by treating it with 100% ethanol. Absolute alcohol. So answer is actually option B. The enzyme used in synthesis of cDNA from mRNA is what? That is reverse transcriptase enzyme. So this is an easy question. Your reverse transcriptase enzyme, what will it do? It will convert your mRNA to cDNA. Okay, now next question, cloning vector with the highest insert carrying capacity is what? So here this was discussed in your lectures. So your plasmid, phage lambda, cosmid, bacteriophage, BAC, yeah. So remember this order. So plasmid has a very less insert size. It has only 15 MB megabase pair. Compared to it, phage lambda has 25 megabase pair, plasmids have 45 megabase pair, bacteriophage has 70 to 100 megabase pair, bacterial artificial chromosome has 120 to 300 megabase pair, yeast artificial chromosome has 250 to 2000. So remember this order. First is your plasmid is the least, followed by your lambda phage. Then this will be followed by cosmids, then bacteriophage. Then you have your back and yak back, and then there is yeast artificial chromosome, which has the capacity to carry the largest DNA fragment. Okay. So now we'll quickly understand this method by which you can prepare cDNA library. So these were covered in detail in the lecture. So we are quickly revising these concepts. Okay, so your Goober-Hoffman method is one method by which you can prepare cDNA from mRNA. So basically, you have your mRNA, and your mRNA has a poly A tail. Your eukaryotic mRNA has poly A tail. So you can create an oligo-DT primer and then amplify that. So that will give you the cDNA. Then you to remove the cDNA, what you do, you treat it with RNase H. So your RNase H will basically it will act on the RNA DNA hybrid and it will cleave the mRNA. So once this mRNA is cleaved, then your DNA polymerase one or the cleano fragment can come and it can synthesize the cDNA or the corresponding DNA, complementary DNA fragment. Okay, and another this so that this is certain your southern blotting, or if you want to probe whether your DNA sequence has that particular probe a particular DNA sequence, you will require radioactive probe. So how do you create radioactive probes? Okay, so radioactive probes are basically oligonucleotide, single stranded oligonucleotide sequences which are having a radioactive group. Okay, so when you're this because it is complementary to your target DNA sequence, it will go and bind to your target DNA sequence, and wherever it will bind, it will give a particular this the you can do an autoradiography, and you from the signal you can identify the band the DNA the particular DNA band. Okay. So how do you do that? You treat your DNA sequence, double-stranded DNA with lambda exonuclease. So it is an exonuclease. So what it does is it this is so in this terminal transferase method, a terminal transferase enzyme will label the probe at the ends to the last nucleotide of the probe. So basically what it has done, it has cleaved a part of this 5' end. So it has cleaved from the 5' end. The probe is incubated with a labeled nucleotide. So you add a particular labeled nucleotide. And, here in this case, in this figure, they have labeled the ATP. ATP with the gamma phosphate of the ATP is radio labeled. Okay, and then you have treated it with terminal transferase. So first you treat it with lambda exonuclease. When you treat it with lambda exonuclease, your exonuclease will cleave a segment of the DNA from the at the 5' end. Part of the DNA sequence will be cleaved at the 5' end. Then you treat it with DATP, radio labeled DATP. So this terminal transferase will transfer this labeled ATP to this 5' end. Okay. So that is so that is the function of terminal transferase, and that is the way you get a radioactive labeled probe. A partial purification with gel filtration column will give labeled primer. So these labeled primers are commonly available for and they are utilized for different purposes. And this is the way by which the labeled radio labeled probes are synthesized. Okay. So Goober-Hoffman method is used for the synthesis of what? It is used for the synthesis of cDNA. Then the type of nuclease enzyme used in terminal transferase method of radioactive probe preparation is what? It is lambda exonuclease. So for radioactive method, first you treat it with lambda exonuclease. It will digest a segment of the 5' end of the DNA, and then you treat it with terminal transferase. Okay, so that the radio labeled DATP can be transferred to the DNA. Okay. So now polymerase chain reaction, you know the four steps of polymerase. There is an initial denaturation, then denaturation, annealing, extension, right? Annealing, extension, and then there is a final extension. And then there is a hold. Hold. Okay, so these steps are repeated 30 times. This is if you have done a PCR, you will see that you can set the number of cycles. So there is an denaturation, annealing, and then extension, right? So the denaturation is done at 95 degrees Celsius, where the two strands will separate, and then in the annealing step, the primer will bind to your DNA, single-stranded DNA, and then in the extension step, your what this Taq polymerase will bind, and it will extend the 3' end of the or extend the DNA in the 5' to 3' direction, isn't it? These are the four steps, and why is PCR required? PCR is required for amplifying your gene of interest. So here also what you will have this formula n equal to n naught 2 to the power n, because exponential 1 plus e whole to the power n. This also is if your efficiency, if you are considering the efficiency of PCR, then you will have this formula. Okay, that also exponential cycle. So component which are not required for performing PCR reaction is what? You require a master mix where you will have this DNA polymerase and DNTPs. You will require template DNA, you will require primer, which are the forward primer, reverse primer. So what are the components required? Forward primer, reverse primer, master mix, where you have the DNTPs and DNA polymerase or the Taq polymerase, but you will not have the DNA ligand. So answer is option B. Initial denaturation temperature during PCR reaction is what? It is 94 degrees Celsius, where your two strands of DNA will get separated. So the vaccine, this was discussed in the last session. Okay, BCG. Now, other than this, there is the restriction enzyme. So the restriction methylase system is what? So this restriction enzyme, why a restriction enzyme present in a bacteria not digest the restriction sequence which is present in its own genome. So the answer is that this restriction enzyme can only recognize those sequences which are non-methylated. So you can see here this the restriction site or the sequence for this EcoR1 enzyme is GAATTC. And here the adenine is not methylated. So this enzyme will act on this restriction enzyme and it will, or this recognition sequence, and it will cleave it, resulting in formation of sticky ends. While here in this case of in the bacterial genome, while the bacterial genome, the adenine is methylated. Okay, so because the adenine is methylated, by this methylase enzyme. So methylase enzyme will methylate all the adenine which is present within this GAATTC sequence. So whenever it is methylated, now this EcoR1 enzyme can no longer recognize the sequence, and this DNA will not be cleaved. So there will be no DNA degradation. If it is methylated and non-methylated, it will be degraded. So there are different three types of restriction enzyme: type 1, type 2, type 3. So you need to remember some of the features of these restriction enzyme. Type 1 is non-palindromic, type 2 is palindromic, type 3 is non-palindromic. The type 1 is why is it not utilized for genetic engineering is because the recognition sequence is not fixed and it acts 1,000 base pair away from where it binds on the DNA sequence, 1,000 base pair away from the site where it binds. So that is why this particular type 1 restriction enzyme is not used, while type 2 is most commonly used. The recognition side, the subunits which are involved, RMS, the cofactor, the product, you need to memorize this table. Okay. So there are different questions that were asked in your assignment. Typical restriction enzyme naming, we all know that it is for genus, species, strain, and the order of identification. E. coli, if you consider. So first they will have the E stands for the genus, which is Escherichia, and then you have the species, which is the coli, and then there is the strain, R, and the order of identification, which is 1, which is the first strain to be identified. So answer is option A. Subunit specificity in type 1 restriction enzyme is provided by what? The subunit, which subunit provides specificity? M is for modification, R is for digestion, You go through the table and the remember the enzyme that are the subunits that are involved. So which type 2 restriction enzyme cuts the methylated DNA? The answer is type 2M, isn't it? Methylated DNA, this RMS, isn't it? Now the type 2 restriction enzyme restriction requires dash as a cofactor. It requires only magnesium, isn't it? So you go through this table very well. Magnesium. Then the restriction enzyme cuts only methylated DNA, but not non-methylated DNA. The answer is DPN1. So this enzyme is very commonly used for site-directed mutagenesis. So during site-directed mutagenesis, when you want to digest the non-methylated or the parental plasmid, so when you treat it with DPN1, so your DPN1 will cut only the methylated DNA. So the parental DNA will be methylated while the amplified DNA that will be non-methylated. So your non-methylated DNA will remain intact while your parental DNA, which is methylated, they will be digested. So answer is option A. What is the start activity of restriction enzyme? So if you keep the restriction enzyme for a very long time, then your restriction enzyme will show non-specific cleaving. Okay, it will not cut at the other than cutting at the recognition site, it will also cut anywhere within the sequence. So that is the start activity. So that answer is restriction enzyme, not self-degradation. Very high efficient digestion of DNA proper, that is for completely failed to cut the alteration in restriction enzyme activity. So answer is option A. It will also show altered activity if you keep the restriction enzyme for a so if the recommended time is that this enzyme is able to digest within 2 to 3 hours, but you are keeping it for 18 hours, 32 hours, then there will be alteration and the enzyme will cut anywhere within the DNA. So non-membrane bound organelle is all you know. You all know it is ribosome, isn't it? SER, RER, these are all Golgi apparatus. These are membrane bound organelle, ribosome and centriole. These are non-membrane bound organelle. Okay, then enzymes which are used in molecular cloning. So these are the sites that I was talking about. So you go through this. So these were discussed in detail. So what are the subunits M, R, S, which site is involved in what? So you see these are the subunits R, S, and F. Is it clear? Then you have what are the difference of these are the different enzymes. Polymerase is used for PCR amplification, which is the main mostly used is the Taq polymerase. Then you have alkaline phosphate. It is required for removal of the terminal phosphate group. DNA ligase is for joining two DNA strands. So ligase, what is the function of ligase? So if your DNA, one strand of DNA has free 5' phosphate group, another strand of DNA has free 3' OH group, then those two strands will be ligated. So it requires a 5' phosphate and a 3' OH. So here in this figure you can see. So there is a 5' phosphate and there is a 3' OH. When you add ligase to it, the two ends will be glued together. But if so when you treat it with alkaline phosphate, your alkaline phosphate basically removes a 5' phosphate group. So when your 5' phosphate is removed, then what will happen? Then even if you add the ligase, your ligation will not take place. Okay, so that is why this alkaline phosphatase has a very useful role in molecular cloning. So this alkaline phosphate treatment enables to enable self-ligation of the vector. So if you do not want if you treat the vector with the restriction enzyme, but after that, what is happening? Your the two ends of your plasmid is getting joined again. So if you do not want that self-ligation to occur, you have to treat it with the alkaline phosphatase so that there will be no self-ligation and only your but so you see here, so this is your gene of interest. So you want to clone your gene of interest in the vector. Now your gene of interest also you have treated it with the restriction enzyme. So the gene of interest will also have a 5' phosphate and a 3' OH. So when you add this gene of interest to your vector and then you treat it with ligase, now because your gene of interest has 5' phosphate group and your vector has 3' OH group, now these two ends will be joined. And when this cloned plasmid will be transformed into the host cell, these nicks that are present, they will be joined in the host cell by the host recovery, the repair pathway. Okay. So it requires ATP basically, or ligase require ATP or NAD plus as a cofactor to catalyze ligation reaction. Ligase is processing ATP to generate AMP. So remember these different points. This complex is binding, will bind to the 3' and 5' of the DNA bearing nick and bring them together. AMP is released, phosphodiester linkage is formed between the 3' and 5'. And so other than this, you have linker molecules, you have adapter molecules. So if you want to clone a gene of interest within a vector, but your gene of interest does not have that particular restrictions site, then what you can do, you can add a linker molecule. So the linker molecule will have that particular restriction site. Then so you can treat the gene of interest, which is to which you have attached the linker molecule. You treat the linker molecule attached to your gene of interest with that particular restriction enzyme, and then you clone it in the vector. So these adapter molecule, then this linker molecule, these were discussed. Okay, you go through it. Now, vectors, components of vectors, what are the good criteria of good vectors? That they should be able to carry a foreign gene. They should have the ability to replicate in the host. Okay, number of properties are desirable. What are the components of vectors? They should have an origin of replication, selectable markers, promoter, right? So if your promoter is not present, then it will not be expressed in the host cell. If your origin of replication is not there, then your plasmid cannot replicate. The copy number will be disturbed. We will get low yield of the plasmid. Okay, low yield of the plasmid, then there will be low expression of the protein as well. So other criteria of growth vectors are that it should have low molecular weight. It should give a phenotypic change. So if it is associated with a phenotypic change post entry, then we can go for selection. Then there should be multiple cloning site and it should have high copy number. Okay, so what are the different vectors available? That is the bacterial plasmid, phage base vector, yeast vector, mammalian vector. So why one vector doesn't replicate in the different host cell? So if you have to replicate that plasmid or that vector within the host cell, you need that particular origin of replication. You cannot add a bacterial origin of replication and then transform that plasmid in a yeast cell and want that it gets replicated. So you have to add a yeast replication origin, ARS, so that it gets replicated in the yeast cell. Okay. So how do you prepare a competent cell? So if you want, you have to prepare a competent cell so that you can insert the clone plasmid in the host cell. So there are different methods. So you remember this list as well. So few questions were asked in the assignment. So for Streptococcus pneumonia, so for bacteria, you treat it with calcium chloride or rubidium chloride or any divalent cation salt. Okay. But for different organisms, you require different other agents. For Bacillus subtilis, you need to treat it with UV light. For Helicobacter pylori, you need to treat it with ciprofloxacin. Okay. So, but for bacterial cells, so how do you prepare competent cells? You do the heat shock method. First, you treat it with calcium chloride, and then as a result, pores are created, and then you do a heat shock method so that the plasmid can be uptaken within the cell. So these are the different steps we had discussed this in detail. So you will so first you treat it with calcium and then you calcium chloride and then you give a heat for 42 degrees Celsius for 1.5 minutes, and then you again keep it on ice for 5 minutes, and then you add the recovery media and then you pellet it down and then you spread it on the LB agar, which is supplemented with the antibiotic. For yeast, you use this spheroplast transformation method where you treat this spheroplast is basically the cell wall, or spheroplast is basically the yeast cell without the cell wall. Okay, yeast cell wall is removed by treating it with zymolase. Okay, And then for DNA delivery in mammalian cells, you have these different methods: chemical transfection method, liposome method, bacterfection, transduction. Then these things are for screening. Screening, we had discussed this reporter gene assay and positive selection, negative selection. So a mutant gene is present, but when you insert that particular, or that the gene is functional, but when you insert that your gene of interest within that gene, your functional gene becomes non-functional. So in that way, you can screen that. So one is this URA3 example. So you go through these slides. Cell organelle known as powerhouse of the cell is mitochondria. This is known. The restriction enzyme cuts only methylated DNA, but not unmethylated DNA. So remember this. This is DPN1 digestion, which is commonly used for site-directed mutagenesis. Okay, for digesting the parental plasmid. Okay, answer is option A. So the restriction enzyme DPN1 specifically cuts methylated DNA because it recognizes and cleaves the sequence GATC only when the adenine within this sequence is methylated. So when this adenine is methylated, only then DPN1 can recognize this particular sequence, or else it will not recognize if it is non-methylated. But the opposite is true for EcoR1. EcoR1 will only cleave the DNA sequence when it is non-methylated. Okay, it will recognize only this sequence, but it will not recognize this sequence. So the T4 DNA ligase enzyme, so bacteriophage T4 lambda, T4 bacteriophage. T4 bacteriophage. T4 bacteriophage is what? It is a virus. So from the T4 bacteriophage, this DNA ligase enzyme was isolated. So that is why answer is it is a it is a virus. It is a virus. Bacteriophage is what? Phage. Phage means a virus. So answer is option A. So UV visible spectroscopy, the shift of absorption spectra towards longer wavelength. So if it is shifting towards longer wavelength, then that is called bathochromic. Bathochromic, longer wavelength is bathochromic. Shifting towards shorter wavelength is hypsochromic. If it is shifting towards higher intensity, it is hyperchromic. If it is shifting towards lower intensity, intensity is decreasing, then it is hypochromic. Okay, bathochromic is when it is shifting towards right or the longer wavelength, bathochromic. So remember these things. Now answer this question. For C-terminal amino acid analysis, the peptides need to be treated with what? It should be treated with carboxypeptidase. Okay, so these different enzymes were discussed. So trypsin will cleave after the lysine arginine. Chymotrypsin will cleave after the aromatic amino acids, phenylalanine, tryptophan, tyrosine. Pepsin will cleave after these leucine, phenylalanine, tryptophan, tyrosine. Cyanogen bromide cleaves after methionine. So remember these different enzymes and where do they cleave? Because they are required for mass spectrometry and for creating peptides that will be used for amino acid or peptide sequencing. Okay, so this carboxypeptide will cleave at the C terminal end. C terminal end means what? At the carboxy end. Carboxy end. Okay. C terminal providing information about the peptide structure and function. C terminal amino acid analysis. Frederick Griffith is associated with the discovery of what? It is associated with discovery of transformation in bacteria. So you remember the transformation experiment, how it was conducted by Frederick Griffith. He took the mice and he injected which bacteria? Streptococcus pneumoniae. Streptococcus pneumoniae. So there were three strains. One is the R2 strains, R2 strain, and one is the S strain. The R2 strain was non-virulent, while the S strain was virulent. So then he mixed the two, and what he found that when the heat killed S3 and the R2 were mixed, there also the bacteria died. Right? Heat killed S3 and the R2. So that is the from the transformation the concept was put forth by Frederick Griffith. So those are all the questions. So you go through the lectures. I tried to cover the important points. Other than this, there were the IEX, SEC, and those techniques, mass spec, IR spectroscopy, UV visible. We have already discussed those concepts in much detail. Okay, so you go through the lectures and the lecture slides, the assignment questions, and also the tutorial sessions. Uh, so that was all for today's session. And So I wish you all the best for your this for this exam of this NPTEL course, and thank you for your attention through the entire course and then today's class.